Today we will cover 8 major topics. First, we'll introduce key concepts about MS. Then we'll discuss the etiology and antigenic targets, followed by non-antigen specific immunity mechanisms. We'll examine viral triggers including EBV, HEV, and CMV. Next, we'll explore the pathogenesis and the role of immune cells. We'll discuss progressive disease mechanisms, lesion pathology and MRI findings, and finally epidemiology and environmental factors. This comprehensive approach will give you a complete understanding of MS from an immunological perspective. Before we dive into the content, let me orient you with our roadmap for the next session. We have structured this lecture into 8 major conceptual domains. We begin with the fundamentals, the introduction and key concepts that frame everything else. We then move into etiology, which is far more nuanced in MS than in most autoimmune conditions. From there, we examine the role of non-antigen specific immunity, followed by a deep dive into viral triggers, particularly the Epstein-Barr virus, human endogenous retroviruses, and cytomegalovirus. The second half of the lecture focuses on the cellular and molecular mechanisms of pathogenesis, the biology of progressive disease, the detailed neuropathology of MS lesions and what imaging reveals about them, and finally the epidemiological and environmental landscape. This is a dense and richly referenced lecture. I want to assure you that every point we cover is grounded in the peer-reviewed literature. The goal is not merely to memorize facts, but to develop a mechanistic understanding of this disease, because it is that understanding which will allow you to reason through clinical scenarios, interpret research findings, and ultimately serve your patients better. Let's get started with section 1. Before we get into details, let me give you the 6 to 10,000 foot view of what the science has established about MS immunology. First, the fundamental fact. MS is caused by immune attack against brain cells. The primary target of that attack is the myelin sheath and the cells that make it, oligodendrocytes, but axons and neurons themselves are also destroyed, particularly as the disease progresses. This is why disability accumulates even when inflammatory activity appears to quiet down. Second, and this is crucial for everything that follows, there is no known antigen that drives MS. Unlike rheumatoid arthritis, where we can point to citrulline proteins, or myasthenia gravis, where the acetylcholine receptor is the target, MS has no confirmed target antigen. No single gene causes it either. This makes it unlike any other autoimmune disease in our textbooks. Third, in untreated MS patients, we see 2 seemingly contradictory things happening simultaneously. Immune control is reduced, allowing unchecked inflammation, and the very interferons that should be coordinating immune defense, specifically interferon alpha and interferon beta, are subnormal. This sets up the immunological dysfunction that defines the disease. Finally, understand from the outset that relapsing and progressive forms of MS are fundamentally different diseases in terms of their gene regulation, immune profiles, and clinical trajectories. What works for one may not work for the other, and this has profound therapeutic implications. We begin with the question of etiology. What causes MS? This is in many ways the central unsolved mystery of modern neurology. We will examine why MS is often called autoimmune, why that label is incomplete, what the candidate antigenic targets are, and how abnormal immune regulation rather than a specific antigen may be the root cause. The etiology section will take us through the candidate antigens, the viral and environmental triggers, and the immune control defects that allow inflammation to persist. Pay careful attention here. Understanding etiology is the foundation upon which all therapeutic reasoning is built. This is one of the most important conceptual slides in the entire lecture. The answer to the question, is MS a typical autoimmune disease, is no, and understanding why it is not is essential. On the left side of your slide, you see the features that are consistent with autoimmunity. There is clearly an attack against self-tissue, specifically myelin and the cells that make it. We see inflammatory T cells, B cells, and antibodies in the lesions. The disease responds at least partially to immunosuppressive therapies. And there are HLA class II associations, which are the classic genetic fingerprint of autoimmune diseases. However, and this is where MS breaks from the autoimmune mold, there is no proven target antigen. This is not for lack of trying. Decades of intensive research have failed to identify an MS-specific autoantigen. The myelin proteins that so beautifully generate experimental allergic encephalomyelitis, or EAE, the animal model, have never been proven as causative in human MS. Furthermore, Mississippi does not arise spontaneously in any animal. You cannot breed a mouse that develops MS. That alone sets it apart. The so-called Goldilocks phenomenon is illuminating. In conditions with excessive type Y interferon activity, like Down syndrome, where chromosome 21 encodes type 1 IFN receptors, or lupus, MS is dramatically underrepresented. Conversely, in untreated MS, IFN alpha and IFN beta levels are subnormal. The interferons have to be just right for immune control, and in MS they are too low but not in the way of classic hyperactivated autoimmunity. The takeaway, MS likely represents an abnormal control of immune responses that permits inflammation in the brain, rather than a textbook antigen-driven autoimmune attack. Despite the lack of a proven causative antigen, a rich body of research has identified numerous candidate CNS antigens that may trigger antigen-specific responses or modify immune reactivity over time. Let me walk you through the main categories. The first category is proteins from infectious agents. The concept here is molecular mimicry, where viral or bacterial proteins share enough structural similarity with brain proteins that immune responses initially directed at the pathogen inadvertently attack the nervous system. Staphylococcal myelinotoxic R3.89 antigen and Clostridium perfringens epsilon toxin are concrete examples of directly destructive bacterial products implicated in this process. The second category, and arguably the most intensively studied, is myelin components. This includes myelin oligodendrocyte glycoprotein, or MOG, myelin basic protein, or MBP, proteolipid protein, or PLP, and myelin-associated glycoprotein, or MAG. Various glycolipids, including ganglioside GD1A, phosphatidylserine, and phosphatidylcholine, have also been implicated. The third category covers glial and neuronal proteins. For oligodendroglia, the most clinically significant finding is antibodies to KIR4. 1. These appear in 50% of both adult and pediatric MS patients' serum and 40% of MS CSF, but in only 1% of other neurological disease patients and 0% of healthy controls. This remarkable specificity makes it a promising biomarker. Also worth noting, alternative splicing of RNA is excessive in untreated disease and is likely generating novel protein targets that we haven't even characterized yet, adding enormous complexity to the antigenic landscape. Now let's examine what we actually find when we measure antibody responses to myelin proteins in MS patients. And the results are, in many ways, counterintuitive. You might expect that if MS is caused by an immune attack on myelin, we should find robust, highly elevated antibodies to myelin proteins, but the data don't support this. Anti-myelin basic protein responses are normal or only slightly increased in MS, a stark contrast to the strong responses seen in the EAE animal model. This discrepancy is one of the core reasons why EAE, despite being an invaluable research tool, is not a perfect model of MS. Antibodies to MBP are actually low in early MS and only increase over time. Detection is also inconsistent between laboratories, which limits clinical utility. Anti-proteolipid antibodies in CSF show interesting demographic patterns, more common in women than men, in patients with later onset or no family history, and with low CSF protein levels. Two findings deserve special mention. First, antibodies to MOG can cross-react with Epstein-Barr virus nuclear antigen, providing yet another bridge between EBV and myelin-directed immunity. Second, heat shock protein 65 is highly conserved between bacteria and humans, and it cross-reacts with the myelin antigen cyclic nucleotide phosphohydrolase. This represents a potential mechanism by which common bacterial infections could trigger or amplify MS. The bottom line, even if myelin antibodies don't cause MS, they may critically modify its course, making them potential disease modifiers and therapeutic targets. We now move into one of the most fascinating territories in MS research, the role of viruses and environmental triggers in initiating and perpetuating the disease. The evidence implicating specific viruses, particularly Epstein-Barr virus, has grown so compelling in recent years that it has fundamentally shifted how many researchers think about MS etiology. We will also examine human endogenous retroviruses, the paradoxical role of cytomegalovirus, the contributions of diet, microbiome, and bacteria, and the emerging role of the aryl hydrocarbon receptor as an environmental sensor that modulates MS-relevant immune pathways. Please note that while individual environmental factors each have modest effect sizes, their interactions, particularly with specific HLA haplotypes, can produce multiplicative risk increases that are clinically very significant. One of the most important conceptual shifts in MS research has been the recognition that inflammation in MS is not solely driven by specific antigen recognition. There are powerful non-antigen specific mechanisms at work. Viruses operate through at least two mechanisms beyond molecular mimicry. They can directly damage oligodendroglia, the myelin-producing cells, causing primary oligodendroglial dysfunction independent of adaptive immune responses. They also have the remarkable ability to enhance the replication of other viruses, making it very difficult to attribute MS to any single viral agent. When we look for the MS virus, we may be looking for the wrong thing entirely. Bacteria and chlamydia in posterior nasal sinuses and submucosa can trigger a cascade: cytokine release, superantigen activation of pathogenic T cells, heat shock protein responses, and direct toxin release. One protective counterpoint, parasite infestation, appears to be protective against MS, a finding we'll explore in more depth in the epidemiology section. Diet affects immunity through both its direct effects on the gut microbiome and by modulating immune cell function directly. Fasting reduces monocyte number and inflammatory activity in animal models. Critically, two bacteria, Acinetobacter and Akkermansia, increase in untreated MS and, when transplanted into germ-free mice, actually worsen EE. The fact that these are countered by high-fiber diets and IFN-B therapy opens the door to dietary interventions as genuine therapeutic adjuncts. The bottom of this slide shows the cascade from inciting event to plaque formation, a sequence we'll revisit throughout the lecture. Human endogenous retroviruses represent a fascinating chapter in MS biology that most clinicians are unfamiliar with. HERVs constitute approximately 10% of the human genome, yet they don't generate complete infectious viral particles. They are ancient remnants of retroviral integrations that have been passed down through human generations. In the context of MS, activated immune cells produce incomplete HERV nucleic acids and proteins. The key finding is that HERV expression correlates with a more progressive disease course, and serum HERV levels rise with increasing duration of MS. This suggests that as the disease progresses, the immune system loses increasing control over these endogenous genomic elements. Two mechanisms by which HERV products may cause damage are particularly well characterized. First, activated astrocytes produce a retrovirus-encoded protein called syncytin, which is directly toxic to oligodendrocytes. This provides a potential mechanism for primary oligodendroglial damage independent of adaptive immune attack. Second, the HERV envelope protein inhibits differentiation of oligodendroglial precursor cells, preventing remyelination even when precursor cells are present. IFN-B therapy strongly reduces the load of HERV products in blood, which may contribute to its therapeutic benefits through this previously underappreciated mechanism. There is also an intriguing clinical observation. HIV-infected patients appear to develop MS at lower rates, possibly because immunosuppression from HIV itself or antiretroviral therapy indirectly suppresses the HERV-driven inflammatory milieu. This is speculative but scientifically plausible and worth monitoring as HIV longevity data matures. If I were to ask you to name the single most important environmental factor in MS risk, the evidence now points overwhelmingly to the Epstein-Barr virus. Let me give you the remarkable numbers. Onset of infectious mononucleosis. The clinical syndrome caused by primary EBV infection in young adults increases the risk of developing MS 32-fold. That is not a subtle association. It is one of the largest effect sizes in the entire epidemiological literature on MS, and critically, the risk is linked to having had symptomatic mononucleosis, not just to acute EBV infection, which affects essentially everyone. The combination of HLA-DRB 115 positive, HLA-02 negative, with high anti-EBNA antibody titers confers a 16-fold higher risk of developing MS compared to the opposite profile. The EBNA2 1.2 allele alone quintuples the risk, and EBV negative MS is extraordinarily rare, suggesting that EBV infection is essentially a prerequisite for developing the disease in the vast majority of cases. So why does EBV do this? Multiple mechanisms converge. EBV-infected B cells produce large amounts of IL-6, which drives B cell proliferation and generates destructive the 17 cells. EBV-infected B cells resist apoptosis and resist immune control, allowing persistent reservoirs of infected cells in the CNS. Viral latent proteins appear in the meningeal germinal center-like areas that are so characteristic of secondary progressive MS-EBV RNA in white matter lesions is associated with interferon alpha production by macrophages and microglia, and perhaps most elegantly, EBNA3 blocks the vitamin D receptor, mechanistically linking EBV to the well-established vitamin D association in MS, as two major environmental factors turn out to share a molecular connection. The EBV story is even more compelling when we look at pediatric MS, because in children, the confounding factors that complicate adult studies are largely absent. Over 90% of children with MS are EBV seropositive, compared to only 50% of healthy children the same age. This is a striking enrichment. The seropositivity to EBNA1 protein increases MS risk 3.8-fold in children, and MS is genuinely rare in EBV seronegative children. One of the most telling findings is about viral shedding. EBV shedding occurs in 50% of EBV-positive children with MS, but in only 20% of EBV-positive healthy controls. This suggests that MS is not merely associated with having been infected with EBV, it is associated with impaired immune control of EBV. The immune system in MS cannot properly contain the virus, and this loss of control may be both a cause and a consequence of the broader immune dysregulation. Now contrast this with cytomegalovirus or CMV. In a finding that at first seems paradoxical, antibodies to CMV correlate with better MS outcomes and lower MS risk. Past CMV infection confers a 0.27-fold reduced risk of MS in children. The proposed mechanism is that CMV effectively exhausts the immune system over the human lifetime. The long-term CD8 T cell commitment to CMV surveillance appears to have a collateral anti-inflammatory effect that is actually protective in MS. This contrast between EBV and CMV, two members of the herpes virus family with diametrically opposite relationships to MS risk, beautifully illustrates how immunological context determines disease outcome. Beyond viruses, several other categories of etiological factors deserve mention before we transition to pathogenesis. Genetics, as a category separate from specific HLA associations, controls the immune response to brain antigens and viruses and also determines the capacity to repair CNS damage. Over 200 genes have weak links to MS, most of them immune-related, and there is massive downstream gene dysregulation that exceeds anything the genome-wide association studies would predict. The role of sex is clinically important. MS is 3 times more common in women than men, and estrogens actively reduce attack rates in women. This sex differential has been increasing over recent decades, from a 2-to-1 ratio to a 3.5-to-1 ratio, suggesting that whatever environmental factors are driving the modern increase in MS incidence are disproportionately affecting women. The oligodendroglia themselves contribute to the etiological picture through intrinsic dysfunction. They exhibit defective function and repair of demyelination, suggesting that vulnerability of the target tissue is not merely a passive phenomenon, it's an active contributor to why damage persists. Regarding vaccinations, contrary to the popular misconception that vaccines worsen MS, multiple types of vaccinations actually tend to reduce exacerbation rates. The herpes zoster vaccines documented 20% reduction in dementia in the general population is an additional benefit relevant to our aging MS population. And briefly, let me explicitly dismiss the CCSVI hypothesis, which consumed enormous research resources in the 2000s. Venous stenting for putative cerebral venous outflow problems showed absolutely no benefit in controlled studies. This episode is a cautionary tale about premature adoption of unvalidated therapies. We now move from the why to the how, the mechanistic pathogenesis of MS. This section is the immunological heart of the lecture. We will examine the 3 phases of MS course, the massive gene dysregulation that characterizes the disease, how T cells enter the CNS, the pre-attack immune activation sequence, the complex cytokine networks including the critically important D17 story, the aryl hydrocarbon receptor as an environmental immune interface, the roles of CD8 T cells, both as destroyers and as essential regulators, and the multiple regulatory cell populations whose dysfunction allows inflammation to persist. This section builds directly on everything we've discussed about etiology. Understanding pathogenesis at this level is what separates a practitioner who can reason about MS therapeutics from one who merely memorizes drug names. The clinical course of MS can be conceptually divided into 3 distinct biological phases, and understanding these phases helps explain why different therapeutic approaches are effective at different stages. Phase 1 is the initiating event. The trigger for the very first exacerbation is unknown. This is where the mystery begins. What we know is that inflammation is triggered by some combination of viral exposure and environmental factors, that brain and hypothalamic lesions form, and that sympathetic nerve damage occurs alongside the CNS inflammation. The initiating event sets off a cascade that, once started, tends to perpetuate itself. Phase 2 is recovery from relapses. This is actually a fascinating phase where multiple restorative processes occur simultaneously. On the immune side, inhibitory TSF2 cytokines rise, glucocorticoids increase, and suppressor T cell function rebounds. On the CNS side, axonal sodium channels redistribute in damaged but surviving axons, a compensatory mechanism that restores some conductance even across demyelinated segments. The brain simultaneously remyelinates and rewires, showing a remarkable capacity for functional reorganization. Phase 3 is chronic progression, the biological phase that remains most therapeutically resistant. Clinical remissions wane as constant low-grade immune activation smolders. Monocyte-mediated damage begins to predominate over T-cell-mediated attacks, and in progressive MS, the cumulative loss of oligodendroglia and neurons increases the demands on the surviving but compromised cells, creating a vicious cycle that current therapies only partially interrupt. Throughout all 3 phases, demyelination and chronic cytokine exposure drive brain atrophy, fatigue, cognitive loss, and neurologic deficits. The clinical manifestations our patients live with daily. I want to pause and emphasize the scale of immune dysregulation in untreated MS because these numbers are extraordinary. When researchers perform RNA microarrays on peripheral blood mononuclear cells, cells that are readily accessible in the bloodstream, they find that 8,800 genes are abnormally expressed in untreated MS patients compared to healthy controls. This is not a subtle signature. This is a massively dysregulated immune system visible in the peripheral blood, remote from the brain lesions themselves. Of these dysregulated genes, the majority are immune-related, and the most dysregulated pathway, more than the Th1 pathway, more than the Th2 pathway, is the IFN alpha/beta pathway. This finding is crucial because it tells us that the interferon system is not just a therapeutic target in MS, it is pathologically dysregulated at the core of the disease. An additional layer of complexity, 3,600 genes undergo abnormal alternative splicing of RNA, largely a different subset from the differentially expressed genes. This means MS is generating novel protein isoforms that may themselves become antigenic targets or may alter immune cell function in ways we haven't fully characterized. The good news in this picture, IFNB therapy largely corrects this dysregulation. When treated patients are compared to untreated patients and healthy controls, their gene expression profiles move substantially toward normal. This normalization of gene expression may be as important to IFNB's therapeutic mechanism as any single measured biomarker. In progressive disease, the gene count drops from 8,800 to 3,600. But now, those 3,600 disturbed genes are in repair pathways, not just inflammatory ones, suggesting the biology has fundamentally shifted. The blood-brain barrier exists precisely to keep immune cells out of the CNS, yet in MS, T cells breach it repeatedly. How does this happen? Following peripheral activation by unknown stimuli, circulating T cells adhere to postcapillary venules in the choroid plexus, brain, and spinal cord. The molecular basis of this adhesion involves integrins on T cells binding to VCAM1 on endothelial cells, which is why natalizumab, an anti-VLA4 antibody, is so effective at blocking this step. Once adherent, T cells penetrate via two routes. They can pass directly through endothelial cells in a process called emperipolysis, essentially being engulfed and transported through the cell, or they can breach the tight junctions of the endothelium after matrix metalloproteinases digest the connections between cells. This step is also blocked by IFNB therapy, which reduces matrix metalloproteinase secretion. Once inside, T cells migrate into perivascular brain parenchyma and form the plaques that define MS histopathology. Early plaques show equivalent numbers of monocytes and T cells, but monocytes progressively predominate in later MS lesions. Critically, brain antigens and immune cells can emigrate in the other direction, traveling to deep cervical lymph nodes where they educate peripheral T cells that then home back to the brain. This bidirectional trafficking creates a self-amplifying loop that is very difficult to interrupt once established. Inflammation, visualized as gadolinium-enhancing MRI lesions, largely resolves within 2 to 8 weeks, but this is deceiving. Unseen by MRI, some immune cells remain in plaques, poised for reactivation. These smoldering chronic active plaques drive the slow, progressive axonal loss that underlies long-term disability. One of the most clinically important insights in MS immunology is that immune activation precedes the neurological attack by several weeks. The brain lesion that causes the relapse is not the beginning of the biological story, it is relatively late in the sequence. In studies that monitored blood immune parameters longitudinally, researchers found that several weeks before attacks of MS and optic neuritis, concanavalin A, mitogen-stimulated IFN gamma and TNF alpha production increases, IFN gamma levels in serum rise, calcium influx into T cells increases, and prostaglandins are secreted by monocytes. All of this in the peripheral blood before the clinical attack is apparent. During the active attack phase, the immune system is in full amplification mode. The activating zeta chain of the T cell receptor is overexpressed on CD4 cells. Costimulatory molecules on B cells increase. Chemokine receptors, CCR5 and CXCR3 on the 1 cells are elevated, and inflammatory cytokines, IL-2 Illinois, 15 Illinois, 17 Illinois, 23 and IFN gamma are elevated in mononuclear cells, while IL-1 Illinois, 6 Illinois, 15 and TNF alpha appear in the CSF. Critically, control of inflammation is completely lost during attacks. Cone-induced suppressor T cell function drops dramatically. IL-10 production falls, removing an important brake on Th1 activity. IL-15 levels rise in blood and CSF monocytes, driving further T cell activation. The clinical lesson: if we could reliably measure these pre-attack immune signatures, we might be able to predict impending relapses and intervene before the neurological damage occurs. Monitoring CD8 suppressor function or expression of CD8, ILT3 or CD80 could help differentiate true MS attacks from fever-induced pseudo-exacerbations, a clinically important distinction. For many years, Mississippi immunology was framed within the T1/T2 paradigm. The idea that pro-inflammatory T1 cells cause disease and anti-inflammatory T2 cells are protective. The reality is considerably more complex. The first complication is that the 1 and the 2 serum cytokines in MS are actually lower than in healthy controls in large-scale analyses. They do increase before attacks in a so-called cytokine storm, but only to near normal levels. They are not chronically elevated in the way the simple Th1/Th2 model would predict. What is abnormal is the balance and correlation between cytokines. In healthy controls, Th1 and Th2 cytokines are tightly balanced and correlated. In MS, they are dissociated. IFNB therapy partially restores this balance. The second complication is the 17 cells, which deserve particular attention. These pro-inflammatory cells amplify autoimmune CNS inflammation and are elevated in secondary progressive MS, correlating with MRI lesion load and brain atrophy. The 17 cells are generated from naive CD4 cells by the combination of IL-6 and TGF-beta and are maintained by IL-23. the inflamed blood-brain barrier, and dendritic cells help polarize naive T cells into the Th17 phenotype. In MS lesions, CD4 and CD8 cells and oligodendrocytes in perivascular areas all produce IL-17. The most toxic T17 variant, cells simultaneously secreting IFN gamma, Illinois, 17, TNF, and GM-CSF, is increased in MS, correlates with MRI-T1 lesions, and critically, these cells are the sole survivors after glucocorticoid exposure. This explains why steroid treatment, while effective for acute relapse, does not provide long-term benefit. It eliminates other T-cell subtypes while sparing the most toxic ones. TAR-17 is particularly relevant in neuromyelitis optica or Davis's disease, the MS mimic, where IL-17 levels are even higher and more pathogenic than in MS. The aryl hydrocarbon receptor represents one of the most interesting molecular bridges between environmental exposures and immune dysregulation in MS. The AHR is essentially an environmental sensor expressed on immune cells. It binds an extraordinarily diverse range of ligands, dioxin and other environmental pollutants, breakdown products of aromatic amino acids like tryptophan, prostaglandins, compounds in cigarette smoke, products of ultraviolet light in the skin, and flavonoids in vegetables, fruits, and tea. When activated, it communicates with STAT transcription factors, retinoic acid receptors, estrogen receptors, and NF-κB, all central to immune regulation. The AHR has paradoxical effects on immunity. On one hand, it can induce inflammatory TH17 cells. On the other, it can also induce regulatory NK cells that secrete IL-10, TR1 cells that also secrete IL-10, and T regulatory cells. Whether the AHR drives inflammation or tolerance appears to depend critically on the specific ligand and the culture conditions, an important caveat for interpreting experimental data. The smoking story is particularly instructive. Smoking products bind the AHR, inhibit NF-κB activation, reduce IFN alpha and IFN beta production, and increase susceptibility to viral infections. Because MS already has subnormal type IFN levels and responses, smoking effectively amplifies this IFN deficit. The result? Smokers have a 60% increase in MS exacerbations, double the rate of brain progression, and double the rate of brain atrophy. The intriguing contrast: oral tobacco containing nicotine actually reduces MS risk, pointing to lung irritation and inhaled combustion products, rather than nicotine itself as the pathological driver. This may work through the AHR pathway, activating pulmonary and systemic immunity in a way that pure nicotine does not. CD8 T cells play a critical and dual role in MS that is not fully appreciated in most clinical training. They are simultaneously the most potent cytotoxic effectors in MS lesions and the most important regulatory cells needed to control inflammation. This duality creates an extraordinary therapeutic challenge. On the cytotoxic side, CD8 clones expand in blood, CSF, and MS plaques. This clonal expansion does not occur with CD4 cells, an important distinction. These cytotoxic CD8 cells directly damage neurons and axons. Many of them appear to be mucosa-associated invariant T cells, so-called MAIT cells, as they react with EBV-infected B cells and secrete toxic IL-17. Melanoma cell adhesion molecule-positive CD8 cells are elevated in the MS CNS and are particularly destructive, producing IL-17, IFN gamma, and GM-CSF, and directly killing oligodendroglia. On the regulatory side, the CD8 plus CD28 negative suppressor cell subset is arguably the most important immune regulatory population in MS. Without its inhibitory feedback, inflammation can become self-sustaining. During exacerbations, the number of these cells drops and their surface CD8 expression diminishes. The suppressor function drops during attacks measured by the concanavalin A stimulation assay in a manner that correlates with clinical activity at a remarkably high level, R equals 0.79. Compare this to the correlation of MRI lesions with clinical activity, only R equals 0.25. These suppressors form a three-way molecular bridge with monocytes to destroy pathogenic CD4 cells expressing HLA-E. The clinical bottom line: correcting CD8 suppressor function is a common mechanism of action shared by IFNB, glatiramer acetate, fingolimod, and ocrelizumab, providing a unifying explanation for how very different drugs achieve similar clinical effects. The regulatory failure in MS extends well beyond CD8 suppressor cells. Multiple regulatory populations are compromised. The classical CD4 plus CD25 plus FOXP3 plus T regulatory cells, TREGs, have reduced function in MS. They can still act through direct cell contact or at a distance by shedding immune inhibitory TNFRI molecules and creating a tissue microenvironment that induces additional suppressive populations, a phenomenon called infectious tolerance. But quantitatively and functionally, they are inadequate to the task. The TR1 subset of CD4 suppressor cells that secrete inhibitory IL-10 is particularly deficient. IL-10 levels are 6-fold lower than normal in MS, and to make matters worse, MS CD4 cells are also resistant to inhibition by IL-10. So not only is less inhibitory cytokine being produced, the cells that should respond to it have become unresponsive. The thymic story is striking and underappreciated. Thymic export of new T cells is reduced in MS. Using recent thymic emigrants, cells whose age can be quantified by T cell receptor excision circles, researchers have found that the immune system in MS is functionally 30 years older than that of healthy controls. An MS patient in their 30s has, immunologically, the T cell repertoire of a 60-year-old. This premature immune aging has profound implications for tolerance, repertoire diversity, and the ability to mount new regulatory responses. CD56 bright NK suppressor cells and follicular regulatory T cells also have reduced function, adding to the cumulative regulatory deficit. Myeloid suppressor cells are the one regulatory population that is actually increased during exacerbations, rising from 5 per million mononuclear cells normally to 140 per million, suggesting a compensatory upregulation of innate regulatory mechanisms when adaptive regulatory cells fail. B cells have emerged as central players in MS pathogenesis, and their biology is considerably more complex than simply producing antibodies that attack myelin. In fact, the remarkable clinical success of anti-B cell therapies like ocrelizumab has paradoxically clarified how multifaceted B cell involvement truly is. On the pro-inflammatory side, activated CSF B cell clones secrete antibodies that generate the oligoclonal bands we find in over 95% of MS patients. B cells present antigens to T cells with exquisite specificity. They secrete pro-inflammatory IL-6, GM-CSF, TNF-alpha, and chemokines. IL-6 from B cells directly enhances the generation of destructive TL17 cells. B cells sequester Epstein-Barr virus and JC virus, creating CNS reservoirs of potentially disease-driving pathogens. And circulating B cells in MS express high levels of co-stimulatory molecules, pushing them above the threshold needed to amplify inflammation. On the anti-inflammatory side, B cells can produce IL-10, Illinois, 35, nerve growth factor, and brain-derived neurotrophic factor, all of which may help repair the CNS. These regulatory B cells, or BREGs, represent a population whose depletion by anti-CD20 therapies may inadvertently worsen the regulatory deficit. This is why anti-B cell therapies will have complex effects. They deplete the pro-inflammatory B cells, but simultaneously eliminate the regulatory ones. The net clinical benefit we observe suggests the pro-inflammatory effects dominate, but the regulatory loss may explain why some patients don't respond as expected and why progression continues even with excellent B cell depletion. The biomarker story, CSF-CXCL13, a B cell attracting chemokine, is elevated in all forms of MS and correlates with CSF B cell number, IgG synthesis, and MRI activity, making it one of the most informative MS CSF biomarkers available. The innate immune system, monocytes, microglia, and dendritic cells, plays roles in MS that are both damaging and, in certain contexts, reparative. Let me walk through each. Monocytes are exceptional antigen-presenting cells in MS. They communicate with cells hundreds of microns away through tunneling nanotubes, literal membrane tubes that transmit calcium ions and antigens between cells. They overexpress receptors for immunoglobulins and are activated even by low levels of serum receptor for advanced glycation end products. Inhibitory molecules on monocytes, HLA-G and ILT3, are reduced in MS but are upregulated by IFNB and by vitamin D and IL-10. Peripherally, monocytes secrete excessive nitric oxide, which is neurotoxic and damages oligodendroglia, but also notably eliminates activated pathogenic T cells. This is one of several examples in MS where the same molecule is simultaneously protective and destructive. Microglia, the resident brain macrophages, release nitric oxide, oxygen radicals complement proteases and cytokines. CSF nitric oxide metabolites correlate with gadolinium-enhancing lesions, clinical activity, and progression. Nitric oxide also modifies brain proteins to create nitrotyrosine, generating entirely novel neoantigens that the immune system may then attack, a mechanism that could explain the progressive broadening of the immune attack over time. Plasmacytoid dendritic cells deserve special attention because they are the major source of type 1 interferons in the body. In MS, rather than producing IFN-alpha and IL-10 as expected, these cells produce less of both and instead induce the 17 cells, contributing directly to the IFN deficit that characterizes untreated MS. The interaction between the neuroendocrine system and the immune system in MS is a clinically important and often underappreciated dimension of the disease. Hypothalamic plaques are common in MS and disrupt endocrine regulation. The periventricular and arcuate nuclei, where regulatory hormone-secreting neurons reside, are particularly vulnerable to CSF toxins and to the lesions that form around the ventricles. MS is unique in having a high number of double-positive neurons co-expressing corticotrophin-releasing hormone and arginine vasopressin, a pattern not seen in other neurological diseases and which appears to reflect chronic hypothalamic inflammation. Since arginine vasopressin potentiates ACTH release, this results in elevated cortisol levels, which may paradoxically inhibit hypothalamic lesion load through negative feedback. The HPA axis becomes hyperresponsive to CRH, especially in primary progressive MS. Chronic HPA overactivity renders cells insensitive to glucocorticoids, which is why high-dose steroid therapy eventually loses its efficacy in progressive MS. The cells have been exposed to chronically high cortisol and have downregulated their receptor sensitivity. The sympathetic nervous system story is equally important. Sympathetic nerve fibers innervate the gut, bone marrow, and spleen and normally inhibit immunity. In MS, the gradual accumulation of spinal cord plaques disrupts these pathways, removing the inhibitory sympathetic tone. The result is a form of denervation supersensitivity in immune cells with overexpression of beta-2 adrenergic receptors on CD8 T cells. This has led to a fascinating therapeutic hypothesis. Oral terbutaline, a beta-2 adrenergic agonist, increases CD8 suppressor function and may have therapeutic potential in progressive disease, work currently being explored. Understanding how relapses resolve is as important as understanding how they start, because the recovery mechanisms inform both prognosis and therapy. On the immune suppression side, recovery is characterized by a rise in inhibitory T2 cytokines, IL-4, Illinois 10, Illinois 13, alongside increasing immunoglobulin levels and glucocorticoid activity. Crucially, subnormal suppressor T-cell function rebounds toward normal as the attack resolves, suggesting that the suppressor defect is at least partially reversible. Inflammation is extinguished by apoptosis of activated immune cells. The one-cell apoptosis is mediated by steroids, both endogenous and therapeutic, as well as by IFN-gamma itself, TNF-alpha, and nitric oxide. IFN-beta specifically causes apoptosis of Th17 cells, which express high levels of the type I interferon receptor. The anti-inflammatory M2 macrophage subfamily contributes by secreting a remarkable array of neuroprotective growth factors, PDGF, EGF, TGF-beta, IGF-1, NGF, BDNF, and neurotrophin-3. On the CNS side, axonal sodium channels redistribute out of the nodes of Ranvier into demyelinated regions, restoring some degree of electrical conduction even in the absence of myelin. This is a genuine compensatory adaptation that contributes to clinical recovery beyond what remyelination alone would predict. The brain simultaneously remyelinates and undergoes functional reorganization, evidence visible on functional MRI as the cortical activation maps change in response to damage. An important caveat: Glucocorticoid treatment, while speeding clinical recovery, has no long-term benefit on disability and actually spares a particularly toxic population of the 17 cells that simultaneously secrete IFN gamma, IL-17, TNF, and GM-CSF. Clinicians should be careful not to overuse steroids based on the misconception that they alter the underlying disease course. A remyelination represents the brain's innate capacity for self-repair, and the numbers here are more encouraging than many clinicians realize, yet also more frustratingly limited. The encouraging part. Remyelination occurs in most lesions soon after lesion onset and continues for up to 6 months. It is the normal default repair response. It occurs in both relapsing, remitting, and primary progressive disease, in both early and late MS. It can be quite prominent in older patients, in long-duration disease, and in subcortical and deep white matter. In 20% of patients, remyelination is extensive. Over 40% of MRI lesions show evidence of remyelination. The cortex remyelinates better than white matter, with myelin sheaths reappearing in 71% of subpial lesions. The frustrating part, in many chronic plaques, despite the presence of oligodendrocyte precursor cells, remyelination fails. The precursors are there, but they don't differentiate and don't wrap axons. Multiple inhibitory factors prevent this. Persistent inflammatory cytokines damage actively remyelinating oligodendrocytes. IFN gamma specifically inhibits remyelination. Jagged 1 on astrocytes activates oligodendroglial notch 1 signaling, which blocks maturation, and a host of molecules including LINGO-1, NOGO, chondroitin sulfate proteoglycans, and hyaluronan all inhibit differentiation. Multiple therapeutic strategies are now targeting this biology. Anti-LINGO antibodies and SIRNA promote OPC differentiation. Clemastine and muscarinic M1 receptor antagonists stimulate differentiation. Ketoconazole modifies cholesterol synthesis and increases sterols favorable to remyelination. Both anti-LINGO and M1 antagonists showed modest but real effects on optic nerve repair in human trials, providing proof of concept that remyelination therapy in humans is achievable. We now enter what is arguably the most therapeutically critical section of the lecture, the biology of progressive MS. This is where our current therapies fail most dramatically, where disability accumulates relentlessly, and where the biological transformation from relapsing remitting to chronic progressive disease occurs. Understanding the transition mechanisms is not merely academic, it is what will drive the next generation of MS therapeutics. The paradigm is shifting from treating relapses to preventing and reversing smoldering neurodegeneration. This section will give you the biological foundation for that shift. We will cover the transition mechanisms, the markers of progressive neurodegeneration, PIRA, progression independent of relapse activity, and the axonal damage cascade that underlies permanent disability. The transition from relapsing remitting to secondary progressive MS typically occurs approximately 10 years after disease onset, though this varies considerably. Clinical remissions wane, constant low-grade immune activation smolders, and monocyte-mediated damage begins to predominate. But why does this transition happen? There are at least 10 distinct biological mechanisms that may drive or reflect this transition, and they interact in complex ways. IFN signaling tells a story of progressive failure, near normal in stable RRMs, subnormal during exacerbations, and continuously subnormal in progressive disease. The interferon system never fully recovers its normal tone. The CD8 suppressor defect transitions from intermittent, occurring only during exacerbations in RRMs, to continuous and permanent in progressive disease. This loss of intermittent regulatory capacity becomes a permanent regulatory vacuum. Naive CD4 T cells that express high levels of activation markers, vitamin D receptor, and toll-like receptors 1, 2, 4, and 7 predict rapid transition from relapsing to progressive MS, suggesting that the phenotype of the T cell repertoire predicts the disease trajectory. Progressive T cell clones acquire antigen-presenting capacity through expression of CD80 and CD86 and become insensitive to steroids and TGF-beta, akin to a pre-leukemic state of autonomous immune activation. Perhaps most troubling, spontaneous apoptosis of pathologic T cells is impaired during clinically active MS, meaning the normal mechanisms that eliminate autoreactive cells are no longer working. These cells persist and accumulate, driving progressive damage. Germinal center-like areas appear in the leptomeninges in secondary progressive MS, organized sites of chronic B-cell activation that continuously emit cytokine toxins onto the subjacent cortex. This meningeal inflammation is now understood as a major driver of the subpile cortical damage that characterizes progressive disease. Continuing our enumeration of progression mechanisms, mechanisms 11 through 16 reveal the full biological depth of this transition. Monocytes and microglia show excessive pro-inflammatory innate immunity in progressive MS, producing 5 to 10 times more IL-12 and IL-18 and more IL-23. These interleukins induce IFN gamma production and allow dendritic cells to activate pro-inflammatory Th1 cells. Meanwhile, pro-inflammatory M1 macrophages and Thyr1 and Th17 cells activate astrocytes to secrete TIMP1 and CXCL10, both toxic to oligodendrocytes. The protective molecules, IL-10, PGE, and BDNF, simultaneously decrease. The shift to innate immunity has a direct therapeutic implication. Anti-T cell therapies, which are effective in relapsing MS, have no or minimal clinical benefit in purely progressive MS. The disease has moved beyond the T cell phase. Neurotrophic failure is another critical mechanism. Nerve growth factor produced by endothelial cells drops with increasing disability. BDNF produced by T cells falls. Chronic stress, possibly including the stress of ongoing brain inflammation itself, reduces BDNF through dimethylation of the BDNF gene, a DNA repressive epigenetic modification. This means the brain's own repair signals are being silenced by the disease. The inexorable damage concept from animal models is sobering. After non-inflammatory damage in mice, remyelination completes and animals are initially stable, but after 6 months, slow axonal degeneration crosses a threshold and clinical function is lost. Residua from old lesions plus ongoing low-grade inflammation combined with aging drive relentless atrophy. Leukocyte telomeres are shorter in secondary progressive disease, suggesting premature aging that correlates with rate of progression. PIRA, progression independent of relapse activity, deserves special emphasis. It increases dramatically with each decade after disease onset, only 1.3% at age 20, but 77% at age 70. Early PIRA is associated with faster overall progression and is the dominant contributor to disability in older patients, more important than relapse-associated worsening in most cohorts. Axonal damage is the primary substrate of permanent disability in MS, and it begins far earlier and is far more extensive than clinical symptoms would suggest. The numbers are startling. MS plaques contain 10,000 axonal spheroids per cubic millimeter, the ballooned ends of transected axons resulting from ongoing anterograde transport, compared to only 2 per cubic millimeter in healthy controls. These spheroids represent axons transected at various times, some days earlier, some years earlier. Early active lesions show 10 to 20% axonal loss. Even in normal appearing white matter, axons are only half as numerous as in control brains. In chronic progressive MS, one half of the axons in many long tracts and the corpus callosum disappear. The mechanisms of axonal damage are multiple. CD8 cytotoxic cells attach to dendrites and axons and transect them by releasing perforin. Monocytes release nitric oxide and glutamate, both neurotoxic. As inflammation subsides, axonal sodium channels redistribute from nodes of Ranvier into demyelinated regions, a necessary adaptation, but one that increases sodium influx. This is accompanied by greater calcium influx, which axonal mitochondria must then sequester. When mitochondria become dysfunctional, calcium-mediated axonal degeneration follows. Low axonal ATP, akin to chronic hypoxia, combined with high sodium and calcium influx, creates a state of chronic energetic crisis. For biomarkers, serum neurofilament light chains peak 8 weeks after gadolinium enhancement onset and correlate with the degree of ongoing axonal damage. CSF tau and neurofilament proteins are modestly better predictors of conversion to definite MS than MRI findings alone. Both increase during exacerbations and in secondary progressive MS, making them valuable longitudinal monitoring tools. Axonal damage predominates in the optic nerve, corpus callosum, and spinal cord. Up to 65% of cervical cord axons can be lost in advanced disease. In the corpus callosum, transcallosal Wallerian degeneration bands on MRI predict poor prognosis. We now shift to the pathological anatomy of MS, what the lesions actually look like, how they form, what cell types predominate, and how they differ by lesion type and disease stage. This section bridges the molecular and cellular immunology we've been discussing with the clinical imaging findings that guide our diagnostic and therapeutic decisions. The Lucchinetti-Lassmann-Bruch pathological subtypes provide a framework that explains some of the clinical heterogeneity we observe in MS patients, and the MRI correlates of these subtypes are increasingly being used to guide treatment selection. We will also cover gray matter and cortical lesion pathology, the biology of oligodendrocytes as the primary target in MS, and the imaging biomarkers that best reflect underlying tissue damage. The MS plaque is the fundamental unit of MS pathology, a well-demarcated area of myelin loss with inflammatory cells, gliosis, and often partial preservation of axons and neurons. Plaques arise in both gray and white matter throughout the brain and spinal cord, but they show a striking predilection for periventricular and periaqueductal sites. Optic nerves are almost invariably involved in MS, making optic neuritis one of the most common presentations of early disease. Cord lesions are often subpial, positioned just beneath the pial surface. Plaques may be preceded by clusters of activated microglia, so-called microglial nodules of 4 to 50 HLA-DR-positive cells in the normal-appearing white matter. These microglial nodules may represent the very earliest detectable lesional change, even before frank demyelination begins. The histological hallmarks are myelin loss, inflammatory cell infiltration, gliosis, and mitochondrial changes reflecting the high energy demand of the lesion. Initially, astrocytes become hypertrophic and gamestocytic with high GFAP expression. When stimulated by IL-9, they produce CCL20, which attracts CD8 and the 17 plus CD4 T cells. Months to years later, astrocytes become fibrillary and form the fibrous scars, the sclerosis, that gives the disease its name. The periventricular location is explained by the remarkably slow blood flow through periventricular veins, less than 50% of other brain areas, which gives circulating immune cells more contact time with the endothelium and therefore greater opportunity to adhere and transmigrate. All MS plaques are perivenular when examined by susceptibility-weighted MRI, a diagnostically useful feature that helps distinguish MS lesions from other white matter disease. The blood-brain barrier is the fortress that the immune system must breach to form MS plaques, and understanding its architecture explains both how it fails and how our therapies interrupt that failure. Structurally, the BBB consists of specialized endothelial cells connected by tight junctions, molecular locks that normally prevent paracellular transit of immune cells and large molecules. On the outer surface are vascular smooth muscle cells and pericytes. The inner layer of the basement membrane includes endothelial cells and pericytes embedded in extracellular matrix. Astrocyte foot processes generate the outer layer of this composite barrier. This is a multi-layered defense system, not a single wall. In MS, T cells breach this barrier through emperipoiesis, being taken up directly through endothelial cells like passengers through a revolving door, entirely bypassing the tight junctions. This pathway is specifically blocked by natalizumab through inhibition of the VLA4 integrin. Alternatively, matrix metalloproteases digest the tight junctions, allowing paracellular migration. IFNB therapy reduces MMP secretion and therefore partially blocks this route. After T cells enter, monocytes follow and pile up in the perivascular space. They then breach the glia limitans, the outer barrier formed by astrocyte foot processes and the basal lamina, using additional MMP activity. The discordance between tight junction integrity and plaque location is instructive. The area postrema lacks tight junctions yet has no increased plaque burden. This tells us that tight junction breakdown alone is insufficient. You also need cellular disruption and activated endothelial cells in the postcapillary venules to actively recruit immune cells. In progressive MS, GD plus lesions are rare, suggesting less acute BBB breakdown. Yet serum proteins extrude more through the BB in progressive than in other forms. The barrier is still compromised, just differently. a slow diffuse leakiness rather than acute focal rupture. Cortical and gray matter lesions represent one of the most clinically significant yet diagnostically challenging aspects of MS pathology. They are common, they are damaging, and they are largely invisible on standard MRI. The Lucchinetti classification of cortical lesions defines three morphological types. Type 1 lesions are contiguous with expanding subcortical white matter lesions. These are the ones we sometimes see on MRI as lesions touching the cortical white matter junction. Type 2 lesions are entirely intracortical, confined to small perivascular areas within the gray matter itself. Type 3 lesions, the most distinctive and the most associated with progressive disease, extend from the peel surface down through cortical layers 3 or 4, creating subpile bands of demyelination along deep sulci. The pathological features of cortical lesions are strikingly different from white matter lesions. There are 10 to 20 times fewer T cells and 6 times fewer macrophages and microglia in cortical lesions compared to white matter plaques. There is less complement activation, less blood-brain barrier breakdown, and minimal edema. This is why cortical lesions don't enhance on MRI. The inflammatory profile is fundamentally different. Despite the reduced inflammatory infiltrate, activated microglia still ensheath cortical neurites and apoptotic neurons, contributing to neurodegeneration through innate mechanisms. Up to 35% of thalamic neurons die in MS. After 30 years of disease, 40% of cortical neurons show synaptic loss. The spinal cord is even more severely affected, 60 to 95% synapse loss and 50% anterior horn cell loss. Type 3 subpile lesions suggest outside-in damage from the inflamed meninges. Soluble toxic factors from meningeal germinal center-like areas diffuse inward, damaging cortex progressively from the surface. This mechanism provides a therapeutic target. Drugs that penetrate the meningeal space and disrupt these germinal centers may be essential for preventing cortical atrophy. Oligodendrocytes are the primary cellular target in MS, and their biology is worth understanding deeply because they represent the nexus between immune attack, myelin damage, and the prospects for repair. Each oligodendrocyte maintains myelin on up to 50 axons, making each cell extraordinarily important for axonal function and survival across a wide network. This is not passive insulation. Oligodendrocytes provide metabolic support to axons, delivering lactate as an energy substrate and producing trophic factors. Loss of even a single oligodendrocyte can compromise 50 axonal segments simultaneously. The sequence of damage is informative. In stressed adult oligodendroglia, myelin changes precedes cell body death. Degeneration begins at the inner cytoplasmic tongue of the myelin sheath, the part most intimately in contact with the axon, and bleb formation precedes cell death. The mRNA binding proteins, TDP43 and PTB, are mislocalized in MS oligodendrocytes, disrupting alternative splicing of RNA and altering production of multiple myelin proteins. Multiple cell types converge on oligodendrocyte destruction. Monocytes and macrophages are the most important quantitatively. Oligodendrocyte loss correlates with macrophage numbers, not T cell numbers. CD8 T cells, NK cells, and gamma delta T cells damage oligodendrocytes through the NKG2D protein. CD4 cells don't directly damage oligodendrocytes but orchestrate the attack by activating the macrophages and CD8 cells that do. The inhibitory factors blocking remyelination are numerous and overlapping. LINGO-1, NOGO, OMGP, MAG, chondroitin sulfate, proteoglycans, hyaluronan, jagged 1/notch signaling FGF receptor conflicts, and declining histone deacetylase activity with age. The complexity of these barriers explains why remyelination therapies have shown only modest effects in human trials. Blocking any single pathway is insufficient when many parallel inhibitory mechanisms exist. The Lucchinetti-Lassmann-Bruch pathological classification of MS lesions is clinically important because it predicts therapeutic response and reflects different underlying disease mechanisms. Pattern 1 affects 18% of patients and represents T cell and macrophage-mediated demyelination. Oligodendrocytes survive and there is rapid remyelination. Lesion borders are sharp. Pattern 1 resembles myelin destruction by macrophage products like TNF-alpha and reactive oxygen species. Theoretically, agents that modify cellular immunity, like interferons, are best suited for this subtype. Pattern 2 is the most common affecting 56% of patients, and adds antibody and complement-mediated demyelination to the cellular mechanisms. Antibodies are bound to myelin, and complement is activated. Plasma exchange has demonstrated specific benefit in pattern 2, but not in patterns 1 or 3, providing therapeutic proof of concept for antibody-mediated pathology. Anti-B cell therapies benefit both subtypes Y and 2, suggesting they have broader immune effects beyond just depleting antibody-producing cells. Pattern 3, in 24% of patients, shows oligodendrocyte dystrophy and apoptosis with specific early loss of myelin-associated glycoprotein and CNP. The distal oligodendroglial processes dying back before the cell body in a pattern reminiscent of acute white matter hypoxia. This pattern suggests a virus or toxin, like nitric oxide, interfering with mitochondrial energy production. 1/3 of MS brains show actual cortical microvascular hypoxia on near-infrared spectroscopy. Pattern 4, seen in only 2% of patients and exclusively in primary progressive MS, shows primary oligodendrocyte degeneration without a clear immune mechanism, suggesting this truly rare subtype may be a primary oligodendropathy with secondary inflammation. The implications for therapy here are profound. Growth factors for oligodendroglial progenitors or actual oligodendrocyte transplants may be needed. A clinically critical observation: active plaques throughout a given brain show the same pathological subtype, suggesting that while patients differ from each other, each patient has a consistent underlying disease mechanism at any given time. MRI is the most powerful clinical tool we have for diagnosing and monitoring MS, but it must be interpreted with a sophisticated understanding of what each sequence actually measures and, crucially, what it does not measure. T2-weighted MRI detects edema. It shows us where water content is increased, which occurs in acute inflammation, demyelination, axonal loss, and remyelination all at the same time. This is why T2 lesions are sensitive but not specific for any particular pathological process. 2 of 9 T2 lesions show no demyelination at all on postmortem analysis, and critically, many T2 lesions are clinically silent. The correlation between T2 lesion load and clinical disability is poor. R equals only 0.2 to 0.3, explaining less than 6% of clinical variance. This clinical radiological dissociation has been a major obstacle to using T2 burden as a primary outcome measure. T1 hypointense lesions, so-called black holes, are much more clinically informative. They reflect actual axonal loss. 80% of T1 black holes are demyelinated. They correlate well with cord atrophy and clinical deterioration in secondary progressive MS as R equals 0.8. Both glatiramer acetate and IFNB reduce the probability that new lesions will become permanent T1 black holes. Gadolinium-enhancing lesions reflect acute blood-brain barrier breakdown and endovascular inflammation. They are most common early in MS and in relapsing disease. 2 or more GD plus lesions predict MS development with 96% specificity after an isolated clinical attack. However, GD plus lesions are only modest predictors of worse clinical course. They should be interpreted in clinical context, not used in isolation to drive therapy changes. The key clinical message: MRI is a tool to support clinical decision-making, not replace it. 2 advanced MRI findings deserve special attention because they are reshaping how we think about MS monitoring and therapy evaluation. Paramagnetic rim lesions and the central vein sign. Paramagnetic rim lesions, also called slowly expanding lesions or SEL, represent one of the most important advances in MS imaging in the past decade. On susceptibility-weighted MRI, these lesions appear as chronic plaques surrounded by a rim of paramagnetic material, iron deposited in activated macrophages and microglia at the lesion edge. This rim persists after the acute inflammatory phase has resolved, reflecting the ongoing smoldering inflammation that we now understand as a primary driver of progressive disability. These rim-containing macrophages and microglia are a fascinating mixed phenotype. They express co-stimulatory markers CD40 and CD86, indicating activation, but also immunosuppressive molecules CD163 and INOS. They remain active for years, slowly expanding the lesion and causing progressive axonal loss. At least one PRL appears in 50% of relapsing and 60% of progressive MS cases, but in only 7% of other inflammatory brain diseases. This specificity makes PRL a powerful MS-specific biomarker. The central vein sign, a thin hypointense line visible at the center of T2 lesions on susceptibility-weighted MRI, is present in over 90% of new and old T2 lesions in MS. It represents the post-capillary venule through which immune cells entered the brain and subsequent tissue remodeling with perivenular collagen 1 deposition. Central vein lesions are more common in MS than in any other inflammatory brain disease, making the central vein sign a potentially important diagnostic differentiator. Therapeutically, PRL are reduced by BTK inhibitors and anti-CD20 therapy. Average progression is slower with anti-CD20 and natalizumab than with interferons and glatiramer, and this difference may be explained in part by differential effects on smoldering lesion activity. Magnetic resonance spectroscopy and quantitative brain atrophy measurement provide important windows into the metabolic and structural consequences of MS that complement conventional MRI. N-acetylaspartate, or NAA, is a neuronal metabolite that reflects the function, though not necessarily the number, of neurons and axons. It is the most widely used MR spectroscopy marker in MS. Early in MS, NAA is already decreased in normal-appearing white matter and in thalamic and cortical gray matter. Long before T2 lesions and atrophy are visible, this early decrease predicts prognosis. The clinical correlations of reduced NAA are powerful. It strongly correlates with fatigue, particularly frontal lobe NAA and fatigue severity, with lateralized cognitive dysfunction, with abnormal visual evoked potentials, and with disability scores. In untreated patients, NAA levels fall progressively. Strikingly, after 6 months of IFNB therapy, NAA in plaques increases back toward normal, suggesting that metabolic disturbances in oligodendroglia, neurons, axons, and possibly mast cells are genuinely reversible with effective treatment. Brain atrophy is the most powerful imaging correlate of long-term disability, but it requires careful interpretation. The atrophy rate in untreated MS is approximately 1% per year, significantly higher than the 0.12% per year in healthy controls, but lower than the 3% seen in Alzheimer's disease. In progressive MS, this rate accelerates to up to 10 times normal. Gray matter atrophies in predictable locations, cortex, especially deep sulci, thalamus adjacent to the third ventricle, and hippocampus at the CA1 and subiculum subfields. Hippocampal volume loss correlates with high cortisol and depression. Deep gray atrophy correlates with cognitive slowing. Cord atrophy correlates best with clinical disability and walking performance. A practical caveat: dehydration for just 16 hours can shrink brain volume by 0.55%, a real confounder in atrophy studies that should prompt standardized hydration protocols before volumetric scans. Our final major section covers the epidemiology of MS, a rich story that has evolved dramatically over the past century and that continues to reshape our understanding of the interplay between genes, environment, and immune system. The epidemiological data on MS are remarkable in several ways. The prevalence has been rising steadily, the geography of risk is striking, twin studies reveal both genetic and environmental contributions, and the specific environmental factors identified, vitamin D, EBV, smoking, obesity, microbiome, diet, provide potential targets for primary prevention. Understanding epidemiology is not merely an academic exercise for neurologists. These data directly inform counseling of patients about modifiable risk factors, assessment of family members, and the design of preventive interventions. Let's proceed. The scale of MS as a public health problem is substantial and growing. The U.S. National MS Society revised the American prevalence estimate to 730,000 in 2019, representing approximately 309 cases per 100,000 people, or about 1 in every 323 Americans. World prevalence has been revised upward to approximately 3 million. The incidence in Olmsted County, Minnesota, one of the best-studied MS populations in the world, has reached 7.5 per 100,000 per year. To put that in historical perspective, over the past 70 years in that same county, prevalence has quintupled and incidence has quadrupled. This is not simply improved case ascertainment. This represents a genuine epidemiological shift. The sex ratio change is particularly dramatic. Over the past 4 decades, the female-to-male ratio has risen from 2 to 1 to 3.5 to 1. The increase in Canada has been documented as occurring predominantly in females. Whatever environmental changes are driving the modern increase in MS incidence appear to be disproportionately affecting women, pointing to sex-specific environmental exposures, hormonal interactions with environmental factors, or immune regulatory differences between the sexes. The increase parallels trends in other immune-mediated diseases, allergy, Crohn's disease, and type 1 diabetes, all of which show similar geographic distribution and increasing incidence. The common thread is altered immune regulation as exposure to infectious agents has diminished, combined with migration to urban environments, reduced sun exposure, dietary changes, and the overall shift from dirty to clean environments that characterizes modern industrialized life. The geographic distribution of MS is one of its most distinctive epidemiological features and provides fundamental clues about etiology. Incidence is highest, greater than 30 per 100,000, in Northern Europe from Iceland to Russia, in Canada, New Zealand, and Southern Australia. These populations share Northern European, particularly Scandinavian ancestry, but they also share high latitude, reduced sunlight, and specific infectious disease patterns. Incidence falls to moderate levels, 5 to 29 per 100,000, in the Mediterranean basin, southern United States, and southern South America, and incidence is low, below 5 per 100,000, across East Asia, India, Africa, the Caribbean, and Central America. Migration studies elegantly separate genetic from environmental contributions. People who migrate from low incidence to high incidence areas before age 15 acquire the higher risk of their adopted country. Those who migrate after age 15 maintain their birth country risk. This age 15 threshold points to a critical window during adolescence when environmental exposures, perhaps viral infections, sun exposure patterns, or dietary factors, permanently set the risk trajectory. The ethnic data within the United States is particularly instructive about how the genetic and environmental relationship has shifted. Decades ago, Black Americans had half the MS incidence of white Americans. By 2010 in Southern California, the incidence in blacks had actually exceeded that of whites, 10 per 100,000 person years in blacks versus 6.9 in whites. The main driver was a dramatically higher rate in black women. This reversal suggests that environmental factors, potentially obesity, vitamin D deficiency, and EBV exposure patterns, can overcome ancestral genetic protective factors when sufficiently powerful. Twin and family studies have rigorously established that MS has a significant genetic component, while simultaneously demonstrating that genetics alone is insufficient to cause the disease. The monozygotic twin concordance rate of 31% is striking from two perspectives. On one hand, it is 200 times the background population risk, confirming strong genetic contribution. On the other hand, 70% of identical twins sharing 100% of their DNA do not both develop MS. This 70% discordance is the single strongest argument in all of MS epidemiology for a major environmental contribution. If MS were purely genetic, monozygotic twins would be nearly 100% concordant. Monozygotic twins carry a 300-fold increased risk compared to the general population. First-degree relatives have a 25-fold increased risk. The sibling risk is 3.5%, modest in absolute terms, but large relative to a background prevalence of 0.1%. When both parents are affected, 9% of children develop MS, and this calculation informs genetic counseling for patients planning families. Mothers and fathers are equally likely to transmit the disease with no evidence of a Carter effect. However, a high-risk mother married to a low-risk father is more likely to transmit MS to a daughter than the reverse, suggesting maternal environmental factors, potentially including microbiome transmission, epigenetic imprinting, or gestational vitamin D levels, play an additional role beyond genetic transmission alone. An important point about familial cytokine profiles: low IL-10 combined with high TNF-alpha in a family predicts a 4-fold increased risk of developing MS and an 8-fold increase in relapsing remitting over primary progressive MS. demonstrating that the cytokine regulatory phenotype is partly heritable and predictive of both disease risk and phenotype. The human leukocyte antigen system represents the strongest genetic association with MS, and understanding the specific alleles involved has both diagnostic and prognostic implications. The most powerful HLA risk factor for MS in Northern European populations is HLA-DRB 11501, commonly called DR2, often occurring in conjunction with DQB 10602. This association is strongest in Northern Europeans, but also present in Japanese and Southern Han Chinese patients with Western type MS. Patients who carry DRB 115 have greater gray and white matter damage and more T2 lesions than DRB 115 negative patients. The susceptibility allele also influences severity. DICA4 deserves mention as it is specifically linked to a primary progressive disease course, suggesting it may predispose to the oligodendroglial subtype of MS rather than the inflammatory T-cell subtype. The risk combination of HLA-DRB 115 positive with HLA-A02 negative in the presence of high anti-EBNA antibody titers confers 16 times higher MS risk than the opposite profile. This three-way interaction beautifully illustrates how genetics and environmental exposure combine multiplicatively rather than additively in determining disease risk. Protective alleles are equally informative. DRB 10901 protects against Western type MS and is found in 30% of Orientals but only 1% of Occidentals, which partly explains the low MS incidence in East Asia. HLA-A0201 and HLA-C05 are also protective. The DR2 and DR4 links suggest two different HLA-linked pathogenic mechanisms for CNS lesions, potentially explaining some of the clinical heterogeneity we observe. Non-HLA-MS genes include immune-related variants, complement factors, cytokine receptor polymorphisms, and a CYP27B1 variant affecting vitamin D synthesis. Over 200 candidate genes have been identified, but individual effect sizes are uniformly small, odds ratios typically around 1.2, meaning no single non-HLA gene substantially predicts individual risk. Beyond the HLA system, genome-wide association studies of thousands of MS patients have identified a rich catalog of single nucleotide polymorphisms that influence MS risk or disease course. These findings are scientifically important, even though individual effect sizes are modest. The predominantly immune-related SNPs tell a coherent biological story. T-cell receptor-related polymorphisms affect how T-cells recognize antigens. The IL-7 receptor alpha chain polymorphism is particularly interesting because IL-7 enhances immunity and affects thymic emigration, and the receptor chain is upregulated by steroids, TNF, and type 1I interferons. The CD58 adhesion molecule SNP has an odds ratio of 0.82 for protection, a protective allele that increases CD58 mRNA expression, potentially enhancing regulatory T-cell function through CD2-CD58 interactions. STAT3 and IRF5 polymorphisms link directly to interferon signaling, the pathway we've repeatedly identified as central to MS immunopathology. The non-immune SNPs are equally informative about the CNS side of the disease. BDNF polymorphisms affect the neurotrophic support available for axonal repair. KF1B affects axonal transport of mitochondria and synaptic vesicles, potentially explaining why some patients lose axons faster than others independent of inflammation. CYP27B, one variation affects vitamin D metabolism, and vitamin D response elements are found in HLA-DRB1-CD40, CXCR4, and CXCR5 genes, suggesting that vitamin D and HLA risk interact at the genomic level. For clinical counseling, the available genetic risk scores have little meaningful predictive value for an individual patient asking about their child's risk. Heritability is distributed across many common variants, each with individually small effects. The cumulative genetic risk is meaningful at the population level, but not actionable for individual clinical decisions. The exposome. The totality of environmental exposures, the exposome, strongly affects MS risk and the interaction between specific exposures and specific HLA genotypes can produce striking multiplicative risk increases. The age of migration data establishes the critical developmental window. People who move from low incidence to high incidence countries before age 15 acquire the higher risk. After 15, migration doesn't change risk. This tells us that whatever environmental exposures drive MS risk, they must act during adolescence to permanently set the immune trajectory. Viral and bacterial infections are both risk factors and modulators. 1/3 of upper respiratory infections trigger an MS exacerbation, with picornaviruses and rhinoviruses being the most potent triggers. Bacterial infections increase exacerbations threefold. Conversely, early childhood exposure to pathogens through infant siblings reduces MS incidence by up to 8-fold, consistent with the hygiene hypothesis. Smoking is one of the most important modifiable risk factors. The relative risk of MS from smoking is 1.5, but when combined with the high-risk HLA profile of DRB 115 positive and HLA-A02 negative, this rises to 14 times the background risk. Smokers have a 60% increase in exacerbations, double the rate of progression, and double the rate of brain atrophy. 41% of new Swedish MS cases with the high-risk HLA profile are smokers. Obesity in teenagers doubles MS risk, and the combination of obesity with the high-risk HLA profile increases risk 15-fold. Adipocyte-derived leptin promotes the 17 inflammation and may represent the mechanistic link between obesity and MS risk. 4 cups of coffee daily may prevent MS development. Alcohol has a strong dose-dependent inverse relationship with MS onset. These epidemiological observations are not merely trivia. They represent evidence-based lifestyle recommendations we can offer patients and their at-risk family members. Vitamin D deserves its own dedicated slide because the evidence for its role in MS is extensive, mechanistically coherent, and clinically actionable, making it arguably the most important modifiable environmental factor in MS after smoking cessation. Mechanistically, vitamin D is profoundly immunosuppressive. It induces CAMP-Illinois-4 and TGF-beta, all anti-inflammatory signals. It inhibits IL-2-Illinois-12 IFN-gamma and TNF-alpha production. It slows proliferation of CD4 T cells, B cells, and plasma cells. It causes a shift toward the 2-cytokine production. And importantly for MS therapy, it enhances IFN-beta signaling, suggesting a synergistic effect when patients take vitamin D supplements alongside IFN-beta therapy. Vitamin D also induces regulatory T cells through an IL-10-dependent pathway. The epidemiological evidence is extensive. Children of mothers in the lowest vitamin D quintile are twice as likely to develop MS. Nurses taking at least 400 IU of vitamin D daily are 40% less likely to develop MS. MRI lesion activity peaks in late springtime, correlating inversely with serum vitamin D levels. Relapses are most likely to occur 1 month after the seasonal vitamin D trough. Tasmanian children with high sunlight exposure are 1/3 as likely to develop MS later in life. The birth month effect is fascinating. More people with MS are born in April, fewer in November, suggesting that maternal vitamin D deficiency during winter months affects fetal immune programming that persists for decades. EBVN3 from EBV blocks the vitamin D receptor, mechanistically linking 2 of the most important environmental factors in MS in a single molecular mechanism and providing a compelling reason why EBV and vitamin D deficiency may act synergistically to increase MS risk. Practical clinical message: vitamin D supplementation is safe, inexpensive, and biologically rational for MS patients. While controlled trial data on therapeutic effect are mixed, the epidemiological and mechanistic evidence strongly supports maintaining adequate vitamin D levels in all MS patients. The gut microbiome represents one of the most rapidly evolving frontiers in MS research, and the data already available are compelling enough to inform clinical practice. 2 bacteria deserve special mention for their disease-promoting effects. Acinetobacter and Akkermansia both increase in the gut of untreated MS patients. When transplanted into germ-free mice, they worsen experimental EEE, demonstrating a causal rather than merely associative relationship. These bacteria are specifically countered by high-fiber diets, which induce competing bacterial populations that produce immunosuppressive short-chain fatty acids. IFN beta therapy also counters these bacteria by increasing short-chain fatty acid transporters, an underappreciated mechanism of IFN beta's benefit. The intestine of MS patients also contains elevated Clostridium perfringens, which produces epsilon toxin. This toxin is particularly dangerous because it binds to endothelial cell proteins and actively breaches the blood-brain barrier, potentially creating a route for CNS entry of inflammatory signals from the gut. The beneficial bacteria story is equally important. Bacteroides fragilis and lactobacillus species help maintain blood-brain barrier integrity, increase regulatory B and T cells, reduce Th17 cells, and control fat storage, obesity, behavior, and even some viral infections. The parasite data are striking. In MS patients with parasitic infestations, clinical and MRI activity is reduced 20-fold. When parasites are treated with antiparasitic therapy, the protection is reversed and MS activity returns, providing a natural experiment that strongly supports the hygiene hypothesis in MS. From a dietary perspective, high-fiber diets counter disease-promoting bacteria and deserve promotion in MS clinical care. The Western diet, high sugar, animal fat, and protein, disrupts immunosuppressive intestinal bacterial populations and should be actively discussed as a modifiable risk factor. Some of the most biologically revealing findings in MS epidemiology come from examining which diseases are unexpectedly rare in MS patients. Cancer is dramatically reduced in MS, by 2/3 to 3/4 compared to healthy controls. This is not simply explained by competing mortality or reduced cancer screening. During clinical trials of subcutaneous IFN beta 1a, the cancer incidence was reduced more than 10-fold in the treatment group, with 50% more cancers in the placebo groups than in the interferon groups. This suggests that either the chronic inflammatory state of untreated MS somehow suppresses certain cancers, possibly through heightened immune surveillance, or that IFN beta therapy has genuine anti-cancer effects, which is consistent with interferon's known role in anti-tumor immunity. Asthma and allergies are half as common in MS patients as in the general population, a finding consistent with the Th1 predominant immune state of MS being opposed to the Th2 predominant state of allergic diseases. The inverse relationship between MS and Down syndrome is mechanistically revealing. Chromosome 21 encodes type 1 interferon receptors, so individuals with Down syndrome have 3 copies, leading to excessive interferon signaling. This high interferon state appears incompatible with the low IFN state of MS. Lupus is similarly underrepresented. Lupus immune cells produce excessive interferon and are hyper-responsive to it. IFN therapy actually triggers lupus exacerbations. The mirror image IFN biology of MS and lupus may explain why both diseases rarely coexist in the same patient. The clinical lesson: few autoimmune diseases are genuinely associated with MS. The presence of apparent MS with associated autoimmune diseases should raise suspicion for neuromyelitis optica, Davids's disease, which is highly associated with autoimmune diseases and has a 10-fold increase in comorbid autoimmunity. Confusing NMO with MS in epidemiological studies creates spurious associations that have led to incorrect conclusions about MS's relationship to other autoimmune conditions. We are now in the final phase of our lecture, and this is a good moment to consolidate what we've covered before we synthesize our conclusions. Looking at the checklist, we have completed an in-depth examination of all 8 major domains of MS neuroimmunology. We've established why MS is not a typical autoimmune disease and why the Goldilocks IFN phenomenon is central to its biology. We've examined the candidate antigens and why no single MS antigen has been proven. We've covered the viral triggers, with EBV as the dominant environmental risk factor, curves as endogenous genomic contributors, and CMV as an intriguing protective factor. In pathogenesis, we moved through the massive gene dysregulation, the 3 phases of disease, T-cell CNS infiltration, pre-attack immune activation, the complex Th1/2/the 17 cytokine network, the AHR as an environmental immune interface, the dual roles of CD8 cells, and the failure of multiple regulatory populations. Progressive disease we examined through 14 mechanistic transitions, including PIRA as the dominant disability accumulating process in older patients, and we examined lesion pathology through the Lucinetti subtypes, blood-brain barrier biology, cortical lesions, oligodendrocyte vulnerability, and the full MRI repertoire. Finally, in epidemiology, we traced the genetic architecture from twin studies through HLA and non-HLA polymorphisms, and the environmental landscape from vitamin D and EBV to microbiome, diet, smoking, and obesity. Now let's synthesize what all of this means. So what do we know and what remains unknown? This slide distills the essentials. On the what we know side, MS is unmistakably immune-mediated. The evidence for immune attack on the CNS is unequivocal. The primary cellular damage involves demyelination, oligodendroglial destruction, and progressive axonal and neuronal loss. Multiple immune cell types are involved simultaneously, and both adaptive and innate immunity are dysregulated. Genetics, particularly HLA, and environment, particularly EBV and vitamin D, determine risk in an interactive, multiplicative fashion. But the what we still don't know side is humbling. The causative MS antigen has not been identified after decades of searching and may not exist as a single entity. The question of why MS doesn't arise spontaneously in any animal and why it's an exclusively human disease remains unanswered. The biological mechanism that triggers the transition from relapsing to progressive disease remains incompletely understood despite many plausible hypotheses, and we still cannot halt progression in the way we can prevent relapses. The failure of current therapies in progressive MS is not merely a drug development problem, it reflects a fundamental biological reality. In progressive MS, the disease has transitioned from an adaptive immune attack that can be pharmacologically modulated to a self-sustaining, innate neurodegenerative process. New therapeutic strategies must target smoldering microglia, meningeal inflammation, remyelination failure, and neurodegeneration simultaneously, not just prevent relapses. The field is moving in this direction. BTK inhibitors, anti-CD20 therapies with better CNS penetration, remyelination promoters, and neuroprotective agents are all under active investigation. The mechanistic understanding we've built in this lecture provides the biological rationale for each of these approaches. This slide provides a framework for connecting the immunological mechanisms we've studied to current and emerging therapeutic strategies. IFN beta therapy targets the deficient type I interferon pathway that is central to MS pathogenesis. By restoring near-normal IFN signaling, it corrects approximately 8,000 dysregulated genes, normalizes cytokine balance, reduces HERV product load, reduces matrix metalloproteinase secretion, and prevents virus infections from triggering attacks. Its limitation in progressive MS reflects the fact that progressive disease has moved beyond the IFN-dependent adaptive immune phase. CD8 suppressor cell correction is a unifying mechanism shared by multiple effective therapies. IFN beta, glatiramer acetate, fingolimod, and ocrelizumab all restore CD8 suppressor function through different primary mechanisms. This convergence suggests that CD8 suppressor restoration may be a necessary, though perhaps not sufficient, component of effective MS therapy. B-cell targeting therapies like ocrelizumab, rituximab, and ofatumumab work through multiple mechanisms beyond antibody depletion. They reduce antigen presentation, Illinois, 6 production B-cell sequestration of EBV, and the costimulatory signals that amplify T-cell responses. The loss of regulatory B-cell function is the main limitation, and future refinement of anti-B-cell therapy may seek to spare or restore BREG populations. Natalizumab blocks T-cell trafficking into the CNS at a very early step in the pathological cascade, before immune activation within the brain occurs. Its effectiveness is consequently very high in relapsing MS. The PML risk requires careful monitoring but is manageable with JCV antibody index testing. For remyelination, anti-LINGO and M1 muscarinic antagonists have shown proof of concept in human trials. The challenge is that remyelination requires addressing multiple inhibitory pathways simultaneously, not just blocking a single molecular target. Combination approaches will likely be necessary. As we approach the end of this lecture, I want to highlight the 10 most essential points that should be your lasting takeaways from today's session. First, there is no proven MS antigen. The core problem in MS is abnormal immune control, not a specific autoimmune attack on an identified target. This fundamentally distinguishes MS from typical autoimmune diseases. Second, the Goldilocks IFN phenomenon. Interferon levels must be just right. MS is characterized by subnormal type YIFN, which allows both increased inflammation and poor viral control. This explains why IFN beta therapy works and why the disease is incompatible with high IFN states like Down syndrome and lupus. Third, EBV is the strongest environmental trigger, a 32-fold risk increase after mononucleosis in young adults, near universal EBV positivity in MS patients, and mechanistic links through IL-6, the 17 induction, and vitamin D receptor blockade make EBV central to MS pathogenesis. Fourth, 8,800 genes are dysregulated in untreated MS mononuclear cells, a massively dysregulated immune system that goes far beyond what genetic susceptibility variants alone would predict. Fifth, the CD8 suppressor defect is the best immunological correlate of clinical activity in MS, with a correlation of R equals 0.79 versus R equals 0.25 for MRI lesions. Sixth, cortical lesions comprise 60% of brain lesions but are largely MRI invisible, making clinical disability MRI correlations unreliable and arguing for more sensitive monitoring approaches. Seventh, progression represents a biological shift from adaptive T and B cell mediated inflammation to chronic innate monocyte and microglial-driven neurodegeneration. Eighth, paramagnetic rim lesions on susceptibility-weighted MRI represent smoldering inflammation, the biological substrate of progression, and are a critical emerging imaging biomarker. Ninth, vitamin D and EBV are mechanistically linked through EBNA3 blockade of the vitamin D receptor, making their combined epidemiological effects more than additive. And 10th, remyelination is biologically possible in MS. It occurs in most lesions early in the disease, but is blocked by multiple overlapping inhibitory mechanisms in chronic disease. Developing effective remyelination therapy requires addressing this complexity rather than targeting single inhibitory molecules. And that brings us to the end of today's lecture on the neuroimmunology of multiple sclerosis. We have traveled from the molecular architecture of the blood-brain barrier to the population genetics of Scandinavian ancestry. From the ancient genomic relics of endogenous retroviruses to the modern epidemiological observation that 4 cups of coffee per day may prevent MS, from the failure of a single regulatory CD8 cell during an exacerbation to the progressive aging of the entire immune system in advanced disease. What I hope you take from this lecture is not a list of facts to be memorized, but a dynamic mechanistic framework, a way of thinking about MS that allows you to reason from first principles. When a new therapy is announced, you should be able to ask which of the pathological mechanisms we discussed today does it target, at what stage of disease, what are the theoretical regulatory cell casualties, can it penetrate the meninges where germinal centers drive cortical damage? Multiple sclerosis is a disease that has humbled many brilliant investigators over many decades. The absence of a proven antigen, the lack of a spontaneous animal model, the failure of our best therapies to halt progression, these are not failures of effort, but reflections of genuine biological complexity. The disease has taught us that the boundary between neurology and immunology is artificial, that environment and genetics are inseparable, and that healing a disease as complex as this will require the kind of deep integrated understanding that we have tried to build together today. Thank you for your attention. I'll now open the floor for questions. No question is too basic or too advanced. These are the conversations that advance understanding